I'm very pleased to introduce Dr. Palau, who is the Head of Genetics and Molecular Medicine Services and Director of the Pediatric Institute of Rare Diseases at the Sant Joan Children's Hospital in Barcelona, where he also directs the Research Institute. So thank you very much for joining us, Dr. Palau, and I look forward to listening to your webinar.
Okay, thank you very much, Tim. So we are going to show here some ideas how in a children's hospital is a third-level university children's hospital in Barcelona, how we are moving in the field of genomics, medicine precision in rare diseases, how moving from genetic testing to genome or genomic testing. And we are going to explain this based in the rare genetic diseases, mainly in the field of neurological disorders, but also immunology and other ones. So this is the next one slide. It's just to present the disclaimers and disclosures, the general one from Illumina. Moving to the next one, just to mention that I have not any disclosure, only to show where is coming the foundation or the funding from different grants, public and private grants, always very competitive grants to support my research group here at the Sant Joan Children's Hospital and Research Institute. And now the 5 points, the 5 main points to guide the webinar today. First of all, I'm going to show the team in genetics at the Sant Joan Children's Hospital, just Children's Hospital in Barcelona, how we have generated our whole services as a unique department, putting together clinical and laboratory services, mainly focused, but not only, in rare diseases. The third point is to think about the new rare diseases or the new medicine landscape from the gene, just the gene, to genome, and looking for different genes and variation inside the exome and the genome, and how to improve rare diseases management and healthcare, the idea to work in the field with a multidisciplinary approach. And at the end, just a few slides to think about the future vision for rare diseases and which is going to be, from my point of view, the role of whole genome sequencing in the field of sequencing and also translational research and focuses to understand the genetic cause of rare diseases. Moving the next one, just to show, to present and introduce my team. Myself, I am currently the director of the genetics department and the Pediatric Institute for Rare Diseases. I'm also the director of the Research Institute at Sant Joan de Déu. I'm consultant at the Clinic Hospital, that's an adult hospital, the major university adult hospital in Barcelona, in Catalonia, one of the major ones in Spain. And also I came from the Spanish Council for Scientific Research, where I am on leave as a research professor. Also I am pediatrician and professor of pediatric at the University of Barcelona. I belong also to the Network of Excellence of Rare Diseases here in Spain that is called IPER. I'm coordinator of Orphanet Spain. This is my group. You can see here putting together the people working in the laboratory, the genetic diagnostic laboratory, people working in the neurogenetics and molecular medicine research group doing genetics, genomics, and also basic cellular molecular biology, try to understand the mechanisms of neurological disorders. The clinical team together here working in clinical genetics, genetic counseling, and rare diseases and undiagnosed disease. And also the bioinformaticians and the microscopy that is working in confocal microscopy at the Research Institute, but also applying functional genomics for the study of specific or to investigate specific mutations that are not really very well demonstrated that they are pathogenic from the genetic analysis and genomic analysis. Now moving the next one, I'm going to show you how we are structured in our clinical and laboratory services. The idea, based on the umbrella of the Pediatric Institute for Rare Diseases, we have the clinical genetics, we have genetic counseling, the gene and genome laboratories, bioinformatics, and also from the Research Institute we have functional genomics, that this is the use of molecular and cellular biology to try to understand from the experimental point of view the pathogenicity of some specific mutations like missense change or variants in candidate genes. So we have the specific also rare and undiagnosed diseases program. Moving to the next one, this is the clinical team, still a small one. We have 3 clinicians, 1 genetic counselor, 1 patient manager, and 1 scientific manager at the group. And now we move to the major outpatient clinics we have. That we have clinical genetics and this morphology that is very relevant in the field of pediatric genetics, genetic counseling, and undiagnosed and rare diseases outpatient clinic. This is the lab team. We have all the people working in cytogenetics, molecular cytogenetics, molecular genetics, and genomics platform, and also the two bioinformaticians, Joan and Gerau. Well, what we are doing now from the clinical cytogenetics and molecular cytogenetics in the laboratory, we are doing postnatal, prenatal, hematological cytogenetics, but also fibroblast culture for clinical investigation. And we're moving to use FISH also for leukemia, specifically childhood leukemia. This is the clinicians or the scientists working in the lab. But also we have implemented quite recently chromosomal microarray analysis that try to do molecular cytogenetics when we are looking for small or duplications or deletions. Now we still remain having quite a lot of techniques, classical molecular genetics techniques, like obvious Sanger sequencing, also MLPA and other kinds of disorders, and a big DNA bank to manage all the samples we are using for research and diagnosis. This is the lab that is conducted by Dr. Loreto Martorell. And the next one is the team working in genomes and genes. We are mainly working at this moment using clinical exome and also in some cases whole exome sequencing. And in the next few years we will move to the whole exome and also to the whole genome sequencing. We have geneticists, we have bioinformaticians, we have the NGS technology, and we are working very close with clinical services try to generate or to looking for correctly the diagnosis based not only in the genetics but also in the contact with the suspicion of physicians when the case is not so clear. At the bioinformatics unit, we have one pipeline that is working pretty well. We have the way to quality control and alignment of the sequences, looking for the indels and SNPs, single nucleotide polymorphism detection, also the copy number variant CMVs detection. And to do this, we have several ways to investigate this, several FASTQ and also several software programs to looking for the aligned risk to put all the sequences together for every nucleotide and to know very well which is the correct nucleotide we have and the possible candidate variant and possible pathogenic mutation in one gene. For the detection of indels and SNPs, we have this pipeline, and starting with many variants when we sequence the clinical exome on the whole exome at this moment. We are using the quality filters, different common variants filter, the genetic and inheritance filter, pathogenicity filter, and then having candidate variants sometimes very clear with changes in the behavior of the predicted behavior of the gene and in a good gene that is very well defined with the phenotype of the disease of the patient. When we are looking for, but not always, and when we need to looking for in depth which variant could be the pathogenic mutation. We are also detecting using exome sequencing. We try to detect with this pipeline CMV that is more difficult when we are using clinical exome or whole exome sequencing. It's easier when it's using whole genome sequencing. So moving from the gene to genome, the NGS, next generation sequencing technology. That is the real situation, and we're going to show you a couple of examples. One case working with clinical exome sequencing, the other one with whole exome sequencing. This is a case, it's a 2.5 years old girl, started the disorder with a non-regenerative normocytic anemia and having a first consultation at the hematology department, and then because she had lymphopenia, especially T cells. Also, there was some consultation to the immunology department, and they try after the conclusion that the patient have non-severe combined immunodeficiency, with looking for using clinical exome sequencing the candidate gene. We found two mutations, the same mutation in the two alleles, at the PNP, that is the purine nucleoside phosphorylase gene, and we observed that the mutation segregating from the father and from the mother. So she was homozygous, biallelic for two mutations or two homozygous mutations with correct segregation. And then we're looking for to confirm, in this case, the possibility by biochemistry studies by HPLC. We tried to looking for if it was really the good candidate. And then in the panel at the bottom, when we change control to the case, we observed that there was one accumulation of nucleosides, adenosine, guanosine, desoxyguanosine, and desoxyadenosine. So we confirm biochemistry studies the finding by genetic, but this is not always possible. We have not always biomarkers to confirm or to propose which could be the gene in the case that we have the biomarker before that, the finding of the gene. But this is a case that we have found in a not very frequent, in a very rare disease. We found the gene because we are moving in this way, and also we have the biomarkers. Doing clinical exome sequencing. Then we move to the next one. This is a 13-year-old girl that shows an overgrowth phenotype since the first years of life, involving tall stature, higher than percentile 97, microcephaly, again higher than percentile 97, and a long and broad hands and feet. The behavioral phenotype was she was a kind girl. She normally used languages to demand an act. She doesn't not present tantrums, and there was not auto or heteroaggressive spells. Love episodes that interfere with her normal activity are not well explained by electroencephalographic findings. And despite she had a delay in speech and developmental delay, she used a wide vocabulary. From the neurological point of view, we have not so much information. There was partial seizures, but the MRI was always normal at the brain and cerebellum. And she had not any gastroesophageal symptoms. That is important because it's quite normal when we are thinking about the gene we found. So we move to west to whole exome sequencing using the projects we have at the laboratory that we are doing exome sequencing based in grants funded by public and also private foundations. What we found at the beginning were a number of polymorphisms, single nucleotide variants, polymorphisms, and filtering by inheritance, we found a number of candidates. Later on, we moved to looking for which candidates could be nuclear pathogenic but not benign. And we found three. The first one, just one example, that the EP13 spline effector variant, internal variant we found, did as a de novo, was also found in other 10 out of 14 analyzed trios at the same time. So it has to be a variant, at least in the Spanish population. And the other two were not evident to be pathogenic. So we moved to looking for, we found three candidates that could be variant, pathogenic variants. And one of them, this is the PAX1 gene, is the gene beta candidate, that is a gene that is encoding a protein having a role and localization at the trans-Golgi network. And then we found one de novo mutation that is an arginine changing to tryptophan at the residue 203. And this was the gene and the specific mutation found in one new syndrome described 5 years ago, that is the Schur-Heumacher syndrome, with a wide spectrum of a neurological level, but also gastrointestinal manifestation that is not typically found, typically but not found in our patient. So we found in our patient this mutation using whole exome sequencing in most of the reading, in 50% of the reading more or less, we found the thymine, that is a change, the variant change from the wild type cytosine. And then we confirm that mutation by Sanger sequencing and a de novo mutation because both the father and the mother show a normal sequence. So the diagnosis was a mutation described in the most of the 20 cases that has been published until now at the PAX1 gene and as a de novo dominant mutation. I want to show now a third case with a different approach. This is a 4-year-old male, son of healthy non-consanguineous parents, The sister were died at the age of 18 due to a possible mitochondrial disease, and he started at 2 years of age due to a frequent falls, foot in the forward truncheons and ataxia, steppage gait, lower and upper limbs reflexes abolished, and normal cutaneous plantar reflex. At the beginning there was found a polyneuropathy, motor and sensitive, mixed axonal and demyelinating. There was an increase, a small increase, very mild, in the creatine kinase. There was even, he was very young, a hypertrophic cardiomyopathy. Also by electroretinography and also ophthalmological examination, there was a retinal dystrophy. And then was proposed to looking for, by genetic studies, genes associated with Refsum disease that were normal. Later on, a panel of Charcot-Marie-Tooth neuropathy that was also normal, and a panel for genes related to hypertrophic cardiomyopathy that was again normal. And after the evaluation, the neurologist thought that could be Friedreich's ataxia, and then he found there was a expansion of the GA repeat, that is a typical mutation in Friedreich's ataxia, but also a point mutation. This is another different case that I published many years ago, but it's a way to illustrate. So the idea in this case is that we have a syndromic diagnosis having neurological expression with ataxia, neuropathy, also cardiac expression with a hypertrophic cardiomyopathy, and also ophthalmologic expression with retinopathy. This is a syndromic diagnosis. So the neurologist was looking for different gene panels, and after evolution and re-evaluation of the case, there was a suspected clinical diagnosis that was Friedreich's ataxia, and finally was demonstrated by genetic testing looking for the 2 types of mutation in the FRDA gene, frataxin gene. What's really happened in this case that is very typical in the medical practice? So the paradigm is that the genetic cause is according to the syndromic symptoms or the diagnosis of suspicion. What really did the neurologist? The physician proposed the genetic cause based on the phenotype. That is normal approach, the classical approach, and is working in many things, but not always. So what really happened in the case that I show you for the immunological case or the Schur-Hoymaker syndrome? It's a different one to this one that I presented to you now on Friedreich's ataxia. There was a systematization of the phenotype. We tried to suspect it from clinical diagnosis, but also we use phenotype ontology, putting ontologies to the specific phenotypes using the human phenotype ontology. Then we did clinical exome sequencing, or mainly in this case, whole exome sequencing, not yet whole genome sequencing. And after, again, phenotypic evaluation and clinical evaluation, we confirmed the genetic study, by genetic study of using whole exome sequencing, the diagnosis of Spurs SHS syndrome. So in this case, the approach is different. The paradigm is that the genetic cause, according to the congruence between the clinical phenotype and the findings in the genome. So the physician, what is doing now the physician, is not proposing one diagnosis or one genetic possible diagnosis based in the clinical suspicion. The physician is contrast the genetic variation with the phenotype. We have a phenotype, we have defined the phenotype better with phenotype ontology systems, and then we move to have genomic information. In this case, in one of the cases, clinical exome sequencing, and the second one, whole exome sequencing. And then we try to put together the phenotype and the variants we found in the exome sequencing. So we are changing the paradigm and the way we are looking for genes and diagnosis, and then what means from the point of view of therapy and also genetic counseling. In the last case of Friedreich's ataxia, we have the classical approach, linear relationship, univocal between phenotype proposing in a progressive way the genotype and then the diagnosis. What's happened in the two previous cases, the immunological one and the syndromic, using exome sequencing, we are having now diagnosis in between phenotype and genotype. So the conclusion we have at the end of our search is in between phenotype and genotype in a non-linear relationship, one-to-one at the same level. Phenotype and genotype are always working together to looking for this last conclusion about which kind of disease has the patient. Now we move to the next one, just to show you some of the results we have in this year, and just to show you that after applying clinical exome sequencing, in all cases we are receiving in the laboratory when we have the suspicion of one rare monogenic or single gene disease, we have a positivity cases with diagnosis, genetic diagnosis, in 59% of the patients we have analyzed until now, after sequencing, bioinformatic approach, a pipeline, and final report. So we need also another approach, not only having the clinical standard approach and the support of the genetics and genomics laboratories, but also to manage it better and to improve the quality of the healthcare we are offering to the patients and parents, but also adult individuals with rare diseases, is the multidisciplinary approach. We have to move in that way. And again, coming back to the structure we have in our Genetics and Rare Diseases Institute. So we are improving the clinical and genetic management. We try to interact very strongly with other clinical disciplines, improving the quality of genetic testing and conclusions, and also solving clinical problems by experimental support doing functional genomics based in molecular cell biology. At the end, we have improving the diagnosis, treatment, management, and counseling, quality of life of the patient and family, parents, and the rest of the family. How we are working at the IPER. IPER means in Spanish, Pediatric Institute for Rare Diseases. Well, we are working with functional plans, but also the day 4. Functional plans means that one specific syndrome, and we started with Prader-Willi syndrome, and now 20q11 microdeletion syndrome. We are working together in few visits at the outpatient clinics in the same day, several days a year, 2 or 3 times a year, with the expert professionals in neurology, genetics, psychiatry, ophthalmology, ear, nose and throat, otorhinolaryngologists, dentistry, cardiology, orthopedics, and other, following clinical guides like the guidelines like American Academy of Pediatrics. We try to know better what is the natural history and the management of this patient, in this case, Prader-Willi. And what is improving really? Families meet together at the waiting room on the same day. There is an increase of knowledge of the natural of the disease for different specialists that they don't know the specialty, what's happened with the specialty that is not specifically for themselves, and also improving the familial and social quality of life of the parents, the patients, and all together. And doing this kind of things is very important to have a patient's manager. So for these functional plans, for this kind of disease, but also they of one specific disease that we put together neurologists and geneticists and clinical manager, we are planning the calendar for the next year. And we have this for Prader-Willi, several days a year, 20q11 microdeletion. We feel that means for microbiota, that is also working in our hospital, Noonan syndrome very soon. And we're moving day off charts, sequence 1q44, X fragile, Soto syndrome, deletion 6p22, Robertson-Taylor syndrome, Smith-Magenis syndrome, Beckett-Wiedemann, Cullen-de-Vries, duplication 15q, Coffin series, Coffin Lowry, SATB diseases, SATB2 diseases, Stickler, Williams, Kabuki, Neil Patella, duplication 20q11, Goldenhar, and a little more. So this is important for the clinical management in a multidisciplinary way and trying to improve the quality of life or the quality of management we have offering to the patient and to know better natural history of the disease. But now having genomics, having the clinical approach based in multidisciplinarity and very close relationship with different clinical disciplines, it's important from genetics, from genetic searching for arriving good conclusion to have always the tree. The familial tree, the pedigree, is very important because we have to think always about inheritance. Not only genetics and epigenetics, but also inheritance. Very important. Because it's relevant for genetic counseling. We are doing studies from genes, from exomes and genomes, try to know better changes that could be the cause of the disease, specifically rare diseases. And then we need to offer better treatments, management, but also genetic counseling. And genetic counseling is something very, very relevant. It's a communication way with good information. And how it's defined by the National Society of Genetic Counseling in the United States. They propose the following definition. Genetic counseling is the process, not only communication, not only information, it's also a process including information, communication, empathy, process of helping people to understand and adapt, not only understand, to adapt also to the psychological and family implication of the genetic contributions of the disease and the condition they have. This process integrates the following aspects. Interpretation of family and medical histories to assess the likelihood of occurrence or recurrence in a family of the disease. Education about inheritance, testing, management, prevention, resources, and research. And finally, advice to promote informed options and adaptation to risk or condition. Well, what is offering in this genomic, clinical, and genetic counseling approach to the rare diseases, which is offering to us rare diseases to understand the way that people became ill and also to understand the nature of human beings. This is a very well-known sentence of William Harvey, more than 300 years ago, thinking about the medicine is there is not any better way to advance the proper practice of medicine than to give our minds to the discovery of the usual law of nature by careful investigation of cases of rare form of diseases, or in modern terms, rare diseases. That way, the translational model we have at the St. Joan de Déu Children's Hospital, in this way for rare diseases, is that the interaction between the patient and family with the physician, or several physicians, managing one rare disease, we have the possibility to make a diagnosis and treatment, but when we have not diagnosis, we don't know the pathophysiology, there is not evident treatment, we need to search for which is the gene causing the disease, and we need to transform the clinical problem in a scientific question. Generating one hypothesis and then moving to the experimental lab, biological lab, and bioinformatics lab, using genomics and other omics like metabolomics and proteomics, experimental research with molecular cellular biology techniques and approaches, and in the future, at the hospital level, looking for gene therapy, small stem cell biology, genome editing, drug repurposing, and then generating new knowledge and new options from the diagnosis and therapeutic point of view to improve the quality of life and prognosis of our patients. And which is the rare diseases future and the role of whole genome sequencing. Some ideas to finish on the future of rare diseases. First one, improving knowledge and awareness of rare diseases and then healthcare pathways and health services for these patients. Better clinical and social management with multidisciplinary teams when needed, and participation of primary care, medical specialists in hospitals, and experts at centers of reference. And then try to put a pathophysiological bridge between rare diseases and common diseases. So looking for new molecular and cellular targets and new therapies. And one way from common diseases to looking for new therapies is investigating in rare diseases that could share pathophysiological pathways with these common diseases. So to offer a global or whole approach to patients with rare diseases based in the idea of genomics, the major biological relevant point of the biology of the individual, that is the DNA, the genome, and genomics, the study of that genome in one specific individual human being, and also to apply to the IRDIC, that is the International Rare Diseases Research Consortium, main objectives in diagnosis and in treatment and in pathophysiology. We try to improve the quality of healthcare management and services, both at the individual and public health approaches, always in patient-centered healthcare, using precision medicine based on the biology and variation of the own genome of the individual. And then improving the quality management and multidisciplinarity of the patients, improving the quality of diagnosis, looking for variants in both health and disease, and pathogenic mutation, and try to get more information about the genome variation of individuals, trying to looking for druggable targets and new therapies, looking for personalized therapeutic targets. So genomics is in the middle, and if we are talking about genomics and the genome, we have to talk about the whole genome sequencing. We want to know not only the sequence of genes, but also the regulatory elements, both promoters, enhancers, silencers, and so on that we have in our genome. This is very important, and this is because I think whole genome sequencing is going to become really very important. Now we know that for a number of limitations we have with whole exome sequencing and also clinical exome sequencing, we have some answers doing whole genome sequencing that we have here illustrated with putting here the haystack, and this is coming from the recent paper published in the Pediatric Clinics of North America by the Clerkman and co-authors. So, conclusions to take home. First one, the topic of rare diseases has become a relevant field in medicine, science, and society. The second one, based on the IRDIC 2017-2020 goals, that is for the next 10 years, diagnosis of all people living with rare diseases is a major objective. To achieve the diagnostic tool, we need to implement genetic testing in the healthcare pipeline and between the whole health system. And last, to carry out these tasks that I mentioned before, it is fundamental and necessary to develop the genomic approach in patient management. And this is already leading us to the application of whole genome sequencing. Thank you very much. On behalf of my team, both clinical and laboratory, all together and research, thank you very much for your participation in this webinar. Bye.
Thank you very much, Dr. Palau. That was an excellent presentation, a very interesting and stimulating presentation that you've given.